Hi everybody, I am Emre Yörük from Istanbul University. The title of the speech is Terpenol is effective essential oil compound product barley from Fusarium avanesium. As introduction, first I would like to introduce Fusarium avanesium, which one is one of the most important casual agent of Fusarium head blight and root rot disease worldwide. This pathogenic species is widely distributed in temperate regions. Particularly, the genome project is released, and it has moderate and high level of genetic diversity. As a barley is one of the most important economically cereal worldwide, and here we see some reports from FAO related to the barley. Particularly in humid and semi-humid regions, are reported for barley to be contaminated with Fusarium avanesium. And fight against Fusarium avanesium has several different strategies, such as disease resistant cultivar development, fungicide treatment, and antagonistic microorganisms. But here we will describe the specific plant derived essential oil compound as terpenolen with the antifungal effects on Fusarium avanesium and not hazardous effects on barley. In material method section, firstly, I would like to introduce our Fusarium avanesium strain AY48 from Russia. It was grown on potato dextrose agar for 7 days. Different concentrations of terpenolen was amended to the PDA medium from 0 to 24 microgram per microliter, and the EC 50 and MIC values were recorded. QRT-PCR assays in Fusarium avanesium. Firstly, total RNAs were extracted by using Trizol reagent, and the common procedure was followed by using a milliliter of Trizol reagent. The quality and quantity of total RNA molecules were checked by spectrophotometer and agarose gels. A commercial kit was used in order to obtain cDNA molecules, and here we see the common protocol for cDNA conversion by Takara, Japan. And in gene expression analysis, beta-tubulin was housekeeping gene, and N-related, catalase-related, and apoptosis-like process-related genes were used as target genes in gene expression analysis. Cybergreen 1-based fluorophore and quantified systems were accompanied in gene expression analysis, and Livak and Schmitzian's formula was used in evaluating the fold changes in gene expression. Here we see two tables related to the gene expression and also CREDRA assays from top to the bottom, from up to the down, in plant materials and electroconductivity and water loss rate assays. Two contrasting barley genotypes, premium and cerevisiae, were used in this study. Potential adverse effects of terpenolen on barley were evaluated via electroconductivity and water loss rate tests at physiological levels. Here we see common procedures for obtaining EC1, EC2, and EC values. Additionally, W24 dry weight and fresh weight values in obtaining the water loss rate values of 14 days old plantlets. In genetics and epigenetics assay, two different methods were used. RAPT, randomly amplified polymorphic DNA method, was used in order to obtain genomic template stability values, and CREDRA assay was used in order to obtain methylation specific patterns alterations. In catalase activity and QRT-PCRs in barley, total proteins were extracted from a gram of fresh leaves of barley samples, and then a common protocol was followed by Harris and Angel. Protein concentrations were detected using commercial colorimetric BCA assay kit, and then the kinetic intervals were recorded for catalase activity. In gene expression assays, alpha actin was used as housekeeping gene, and WRKY6, 24, 25, and 41 genes were used as target genes. In statistical analysis, 2-case LCD test, a one-way ANOVA student tests were used, and heatmapper CA was also used in obtaining heat maps. As a result, here we see firstly EC50 value was recorded as 3 microgram per milliliter terpinolone treatment, and in gene expression assays high quality and quantity of total RNA molecules were extracted, then they were used in gene expression assays, and we see no significant differences for Xenon synthesis rated gene. However, N-niacin synthesis and auroxorin synthesis rated gene expression were significantly altered. In Barley tests, electroconductivity and water loss rate assays, here we see EC values and also water loss rate values were not significantly different from control sets in experiment sets. Similarly, in genetics and epigenetics assay, low level of genomic in template instability was recorded, with the genomic template stability value of 74 and 85 and 75% in cervoise and premium cultivars. Similarly, 20.6 to 12.5% methylation differences were recorded in Credra assays. In addition to electroconductivity and also water loss rate assays, catalase activity and QRT-PCR analysis were also showed that terpinolone did not significantly change the expressions of two barley cultivars. Here we see table for fold changes in gene expression, heat map assays with no significant changes, and idiomorphic and homomorphic bonds from Credra assays. In discussion section, we could easily say that we know that several different strategies are used in fight against Fusarium avineum, and one of the most important strategy was fungicide treatment. But high level of genetic diversity and unconsciousness in fungicide usage led to the increasing resistance to common fungicides such as thiophanate methyl and carbendazim. Previous reports published in the last 2 decades show that the plant-derived essential oils could be alternative approach to fungicide treatment, and in the study we have shown that. Here we described potential antifungal effects of terpinol on Fusarium avineum, which one is one of the most important causal agent of head blight and crown rot disease of cereal, all small grain cereals worldwide, and terpinol on not affecting characteristics of terpinol on barley for the first time. And we can say that we obtained relatively low level of MIC values for terpinol treatment in comparison to previous studies. N-niacin synthesis rated gene expression was significantly downregulated, such as in previous studies, and further studies could include terpinol treatment in vitro and implant studies to fight against Fusarium related diseases. And further studies could also include plant tissue culture tests and mammalian cell line tests. Thanks for listening me. Please see the www.phytopathology.com and you can download this PDF file of the speech.